My name is Howard, and I'm going to present our physics project. Here are my group members, which are Helen Stan, Yavan, and Hui Qing. We all are from Pro S1 class. Now we look at the acknowledgement. We would like to express our profound appreciation to all of those who provided us the opportunity of completing this report. A special thanks goes to our physics teacher, Juan Norani, who contributed in providing suggestions and encouragement. He has helped us manage this project, especially in making this presentation. Furthermore, we would like to acknowledge with much appreciation to crucial role of the lab assistants who permitted us to use all the required equipment and the necessary materials and needed to complete this assignment, which is the use of lenses in optical instruments. Props to our group members who helped assemble separate aspects of the assignment and who gave practical suggestions too. Finally, we are grateful for the guidance given by the other supervisors as well as the panels, especially in our presentation. Our presentation skills have no doubt improved thanks to their constructive comments and advice. Now I pass the mic to Wei.
So as an introduction, the term lens is applied to a piece of glass or transparent plastic, usually circular, that has two surfaces that are ground and polished in a specific manner designed to produce either a convergence or divergence of light. The two most common types of lenses are concave and convex lenses. A convex lens or converging lens is a curved lens in which the reflective surface bulges towards the light source. Convex lenses reflect light inward to one focal point. They're used to focus light, and such lenses form either a virtual or real image, unlike concave lenses. Convex lenses show different image types depending on the distance between the object and the lens. A concave lens, or diverging lens, has a transparent surface that is recessed inward, away from the incident light. Concave lenses refract light outward, therefore they are not used to focus light. Since the focal point F and the center of curvature 2F are both imaginary points that cannot be reached, image formed by these mirrors cannot be projected on screen. The image is smaller than the object but gets larger as the object approaches the mirror. Microscopes vary in type and power, but the most common type uses a combination of lenses to magnify an image. This allows one to see things that one wouldn't be able to view with the naked eye. Our objective is to study the use of lenses in our daily life, to understand the uses of optical instruments, mainly a simple microscope, to create a model of a microscope, and to learn how to use microscopes to produce an image of an object in which the two components imaging the objects are the convex lenses. So the materials we use to make our model is two convex lenses, 3 toilet paper rolls, super glue, which is Yoo-hoo glue, black paper, and white paper. And our apparatus is ruler and scissors. So this is how we made it. First of all, the distance between the two lenses where a clear and magnified image of the object is measured and marked. In this case, it's around 21 centimeters. The first two toilet paper rolls are cut lengthwise accordingly, where the total length of the two will be equal or slightly lesser than said distance. The diameter of the first two toilet paper rolls are resized to be smaller than the thirds. Super glue is applied in between the layers, the inner layers to solidify a structure. The first roll is wrapped with as many layers of white paper as needed to fit snugly in the third roll. So this is so its position can be adjusted to change the distance between the lenses. The same is done for the second roll, but with black paper. The third roll is wrapped with a layer of black paper, and using super glue, each convex lens is adhered to one end of each of the first two rolls. The free end of each roll is inserted into both sides of the third toilet paper roll. So I'll show you the model right now. We see a model over here. This is the eyepiece. You look through the eyepiece here. It looks like a telescope, but it's a microscope, and it's adjustable, so the distance between the lenses can change. This is the objective lens. This is the eyepiece. And the microscope is also tested to see if you can actually see a magnified image. So there's some safety precautions to be aware of. Be careful when using scissors, handle super glue with care, and handle the lenses with caution as not to drop them. I actually chipped one of them over here, if you can see, because I did drop them. And here's a sketch of them. This is the front view. You can see the eyepiece here and objective lens. And here are the measurements. So the whole thing is about 22 centimeters long. The middle part is about 10.5 centimeters. These are black and this one is white. So here's the eyepiece, here's the objective lens. I'll pass the mic to my teammate to finish it off.
Light passes through the specimen and into the objective lens, which the project are real in water and magnified image of the specimen to fixed plane with the microscope. This happens through the refractions of light. The objective gathers the light coming from each of the various parts of the specimen. The objective must be positioned by the user so that it will be focused close enough to the specimen, so as to project a magnified real image up into the body tube. The eyepiece or ocular, which fits into the body tube at the upper ends, is the farthest optical component from the specimen. The placement of the eyepiece is such that its eye lens further magnifies the real image projected by the objective. The eye of the observer sees this secondarily magnified image, which appears as if it were near to the base of the microscope. The eyepiece serves to further magnify the real image projected by the objective. An important feature of microscope optical component is their very short focal length that allow increased magnification at a given distance when compared to an ordinary hand lens. As the focal length of the lens used are relatively long, the magnification power of the microscope is not as powerful as a regular microscope. And this is the conclusion that we get. The use of optical instrument, for example, a magnifying glass or more complex device such as a microscope or telescope, usually makes the thing bigger and help us to see in the more detailed manner. The use of converging lens makes things appear larger. On the other hand, diverging lens produce a smaller image. Convex lens focus distant image at a point on the side of the lens opposite from the light source. When the parallel rays of light passes through the convex lens, the reflected ray converge at a single point called the principal focus. Inversely, concave lens focus distant objects, so someone on the side of the lens opposite to the incident light see the object as to be at a point behind the lens. When parallel rays of light passes through a concave lens, the reflected rays diverge, appearing to come from one point called the principal focus.
All right, so this is a few references, a few links. These are the materials used. You can see toilet paper rolls, convex lenses. And this is our first part, second tube, this is the middle tube, and they're assembled like this. And here's an example of the usage. You can see the fibers of a bed sheet here. See them up close. That's all for our presentation. Thank you everyone for listening.